Hello, and welcome to today's NCBI Minute on our updated ORFfinder, an open reading frame prediction tool. I'm Wayne Mattin. With me today is Peter Cooper, and you can see on this slide the closed captioning information. Go ahead and put in your questions in the question pod as you think of them. Peter may very well be able to answer those while I'm talking, but we'll put those in a document, and any that we get after I quit talking into a link in materials on the webinars and courses page, and I'll just go ahead and show you that page. So this is webinars and courses. This is today's webinar, but that will, before very long, move over to the archived page, and there you see a link in our previous webinars to materials, so those questions and answers will be there. There are no slides from today. Eventually there'll be a video file in materials, and we'll also post a recording on our YouTube channel. Probably get that up within the next several days. If you have questions about ORFfinder after we stop the webinar, feel free to go ahead and email me at Matten, M-A-T-T-E-N, at ncbi.nlm.nih.gov, or you can send general NCBI questions to that info email alias. So this is intended to be a very short minute, so let me go ahead and jump over to the ORFfinder page. You can find this in a web search if you search for NCBI ORFfinder. You may well come upon the old ORFfinder page, but there's a link there to this page. And the URL is simply the base NCBI homepage URL /ORFfinder, all lowercase. So what's new in ORFfinder now? Primarily it's the interface. It looks more like many of our other resources. You'll see that particularly on the results page. But we do now also have a standalone version. You can download that binary here at this Linux x64 link. And the standalone version does not have the limitation of processing 50 KB that the web version does. The other addition is you can now send your translated ORFs directly to SmartBLAST, and we still maintain the ability to send it to the full BLAST-P service as well. Just a quick caveat before I demonstrate, ORFfinder is not intended to be, a gene prediction tool, particularly for eukaryotes. It works in many cases fine as an ORFfinder in bacterial and archaeal genomes, viral genomes, and also for eukaryotic transcripts. I'm not going to demonstrate the transcripts today, but there's a link here to demonstrate that for you if you want to look at that. What I'm going to do today is enter an accession for an archaeal genomic sequence. There's that RefSeq accession. This sequence is actually close to 3 million nucleotides long. I could put in some coordinates here in from and to, but ORFfinder will automatically stop the processing at about 50 KB, so I'm not going to bother with that. You have a few options here. I'm going to go ahead and select a slightly larger minimal ORF length just to clean up my results a bit. You may or may not want to do that depending on your goals and your sequences. I know I'm looking at an archaeal sequence, so I'll go ahead and select genetic code 11. And I'm going to choose to include alternative initiation codons. You may, in some cases, want to use this AnySense codon option, particularly if you have a feeling that your sequence may not begin at the beginning of the open reading frame. So if you choose AnySense codon, you'll be able to perhaps walk upstream to a different initiation codon. Okay, and I'm going to go ahead and check ignore nested ORFs again, primarily just to clean up my output because this is a fairly long input sequence. I'll click the submit button, and this is running in real time. This is very fast. And given the conditions I had, it found 115 ORFs, and it reminds me that it calculated that on the first 50 KB. I can also notice that by sliding over here in the viewer and see that the processing stopped right about 50K. Okay, so those of you familiar with our gene records and our genome browsers will recognize this graphical viewer as our sequence viewer. And so it has the same controls for zooming, very similar list of tools, and I'll point out a couple of those later that you might like. And then below that graphical view is a table that's sorted initially by length of the ORF, so the longest ORF's on top. But these columns are all sortable. So for example, you might want to see the ORFs in the order on the input sequence strand, or input sequence. So I'm going to go down here and select ORF46. It's the first longish one. And when I select that, it then gets highlighted up here with a marker in the sequence viewer. Now many of you are used to using a 6-frame translation in ORFfinder. To do that here, you click this button, Add the 6-frame translation track. Because this is sequence viewer, it adds that track. And I'm zoomed out too far to see any detail here, so let's go ahead and zoom in on this first large ORF, ORF46. I'll mouse over that marker name, and that gives me the option then to zoom to sequence marker. So I'll click that. And now I'm zoomed all the way into the sequence. The shading here represents the beginning of ORF46. And you can see that in fact it used a CTG as an alternate initiation codon to begin the ORF. And down in the table you can see that this is in frame +3. So let's take a look at the 6-frame translation +3, and we see here's a ATG methionine, and down just a little bit farther another ATG methionine. And so that would be a good use of BLAST perhaps to see if we can get some information about what other related proteins actually start with. And we'll do that in just a minute. But first, let's look at some of the other options we have on this results page. So you notice a button here that's grayed out at the moment that says Download Marked Set. So I could mark all 115 of these ORFs with this button, or I can pick and choose ORFs. So how do you mark them? You have one selected here. You're looking at the translation here in the box on the left. Then you can mark that one that you're viewing. So that changes the color in the table, and it actually puts a little gray box around the ORF up in the viewer. So let's say I want to select now ORF47. I click on that in the table. Now it's being displayed on the left, the translation is, and then I can mark that one as well. So now I have 2 marked ORFs up in the sequence viewer. And so now my button to download mark set is active, and I can download then either FASTA, ASN1, or a feature table format. You'll notice here that's grayed out, you can't select it at the moment, is CDS FASTA. That's because this is not implemented yet, but hopefully it will be before too long, and that will allow you to download the nucleotide FASTA for that coding sequence region. Okay, and marking these also allowed us to use BLAST marked set. So these 2 buttons will send the translations off to the standard BLAST-P page. You can select a database here, but you get to the full page where you can select other databases as well, and you also have all the algorithm parameters you can play with. I'm just going to demonstrate SmartBLAST. SmartBLAST does not have a SmartBLAST the mark set button because SmartBLAST will take only one query sequence at a time. So if you do want to analyze multiple ORFs at once with BLAST, use the full BLAST-P algorithm. I'm going to go ahead and unmark this ORF47. So I have this up here. I can click unmark. Now I have just one of these marked, but I want to BLAST 46, not 47, so I click on that again in the table, and that loads ORF46 up here. So the question is, which of these methionines might be the actual one that's used as the initiation codon? So note those up here, and I'll go ahead and click on SmartBLAST. And again, this is also running in real time, and it's finished. If you're not familiar with SmartBLAST, we had a webinar on that back in September, so you can check that out on our YouTube channel. This did in fact search the NR database, but it uses a faster algorithm, so that's why it comes back so quickly. Search is another database we called Landmark with a much smaller set of sequences, but that's detail we don't need to worry about today. And we got some names here, which means we probably got some pretty decent hits. So I'm just going to mouse over that name and click on alignment in that pop-up menu, and that takes me down the page to the alignment section. So this in fact is a self-hit. This is the genomic piece that I put in for this particular strain. And we can see here, this is actually that second methionine, the first ATG methionine. And then if we scroll down a bit, here's another species, uses the same methionine. And scroll down to another one, yet another species of the same genus. It's also using that one. So that's some evidence anyway that it uses that first ATG as the start of that particular coding region. All right, I'm going to go back to ORFfinder. I have just one more example here. So I'll go back to the submitting page. And here I'm going to input an accession for a bacteriophage. And then this is a much smaller sequence. I'm going to just keep all the defaults and go ahead and click submit. It found 21 ORFs this time. And I just want to point out, so if I had checked the box for ignore nested ORFs, I would have seen this one, this one, this one, and this one. So I would have gotten 4 ORFs. It would have ignored all these smaller ORFs that are completely enclosed by a larger ORF. And we're not going to do any analysis here. I'm at about over 10 minutes already, so I'm going to wrap this up pretty quickly. I just wanted to point out, since you're in the sequence viewer, there's some nice features here. For example, you could download a PDF version of this graphics view. You might want to get rid of the label first on this ORF by removing that marker. And if you wanted to send a link of this view to a colleague, you could go over here to the pull-down menu next to the question mark and click on Link in View. Tells you that this link will be active for 1 to 2 months. And then you could send your colleague that tiny URL there. Okay, that's it for this quick demonstration. I'll remind you, if you have any questions, you can write to me or send those into info. Do we have any questions online, Peter?
No, none right now.
All right, I will close the webinar then. Be sure to check out our courses and webinars page and our YouTube channel to find out more about what we're doing. Thanks. Goodbye.